So the way I'm going to organize the review session is first I'm going to open it up to any questions that you guys have. I'm also going to go over questions I got in the feedback survey from this week. I think I already went over everything from last week. Um, and then um, I'm I'm happy to end the review session, or if there's just kind of general topics that you would like me to go over but you don't have any specific questions, I can also go over that as well. So I'm just going to start by asking if anyone has any questions for me about the exam. So if you have no specific questions, were there any part of the was there any part of the material in chapters 1, 2, and the microscopy that we did and that you read about in chapter 3 that was challenging? Or any questions from the homework that you didn't understand the answer? You should be able to review those by going back into that assignment and hitting the review button. That's usually on the very right of the screen. And otherwise I can open it up to, I can start with the questions that I got from the feedback survey.
I have one kind of specific question
Yes, Mike.
on the concept of invasiveness. The definition the book gives says ability of a bacterial pathogen to rapidly spread through tissues. Does that preclude viruses? Would they be described differently?
Yeah, that so generally that's used for bacterial or microbial pathogens that are living, so microorganisms like fungi, because viruses don't have an invasive trait. They have, we use different terms to describe their pathogenesis. For viruses, they can only go to the cells that are able to recognize them. So, for example, influenza virus is very limited in its scope because it only infects the epithelial cells in our upper respiratory tract, whereas something like Ebola has many different cell types that it in various tissues that it can infect, and so it's really a systemic symptoms that we get from that infection. So it's not that it's more invasive. It's simply that it has a different spectrum or repertoire of cells that it can affect.
That makes
Whereas
sense. It doesn't
bacteria,
necessarily preclude, it's just based on the nature of the way viruses work.
yeah, they work differently. So still, bacteria still have to recognize and attach molecules on the cells in our tissues in order to infect. Some of them don't invade. Some of them simply colonize the surfaces of our body to cause infection. Others have mechanisms that change the physiology of our host cells and enable them to enter. So for example, something like E. coli can make us sick. It's a gastrointestinal infection, can give us diarrhea. So can Shigella. They're both bacteria. They both require attachment to host cells. But E. coli stay in the lumen of the gut, and they colonize the surface, the epithelial cells that line that lumen of the gut. Whereas the Shigella, they attach to the cells lining the gut, and then they are able to use a system that we'll learn about later in the semester to introduce bacterial factors into the host cells that then change their physiology, and they kind of engulf the Shigella bacteria and bring it into the cell, and it's able to invade the tissue in that way.
Gotcha.
Yeah.
So to your to the way you kind of gave that answer and gave a couple examples of certain microbes that exhibit this characteristic or that characteristic, I noticed the book definitions also tend to do that. Whether it's mode of transmission or method of infection, whatever it may be, but there's often an example given. Will the exam kind of go to that level, or are we looking more at concepts like know how these work, but you don't have to necessarily name the one that the book says that does it?
Okay, yes. So how much detail about these definitions and these concepts in Chapter 2 do you have to know? So you have to understand invasiveness. If it's helpful to know the example that they give in the book, then by all means, study it. You don't have to memorize it. I'm not going to ask you about any specific types of microbes or microbial agents. I more want you to understand the broad differences, the general differences in the groups, such as, the difference between a virus and a bacterium, rather than what kind of bacterium causes gastrointestinal illness, or
Right.
as an example of an invasive bacteria. We'll learn that along the way, but I'm more concerned with you getting the concepts. So as it mentioned in the beginning of chapter 2, this is a lot, the chapter 2 is a lot of epidemiology. It's very relevant to specific diseases, but we haven't learned the specifics yet. We're just learning these concepts so that when we do go back and learn about the structure and the function of these variety of microbes, that we'll have a context in which to think about it and learn about it.
Makes sense.
So for the test, you don't need to know bacterial names. I will use some bacterial names, um, but it's more like one of the. Let me share with you the. Let's see, Google Chrome. Yes. Okay. So I'm going to share the screen with you so that you can see the study guide that I have up. So on the study guide, for example, under the possible topics for questions that are short answer or multiple choice or true false, I have a question like, what is the binomial system used to name bacteria? So I would give you a name and ask you to explain, well, how does the binomial system work? What is the first name? What is the second name?
Right.
What is a genus? What is a species? Those kinds of things.
Okay, makes sense.
Okay? Okay.
And similarly with some of the microscopy stuff, theta angle, equation for the refractive index. My guess is that's a little deeper than we need to go, right?
Yeah, definitely deeper than you need to know. So probably I'm expecting that you've already learned about microscopy and this should be review, but you should absolutely know the items that I've listed on the study guide here in the middle of the possible topics list. How to magnification, so you need to know what magnification is, and contrast, you need to know the definition or what that is describing about microscopy. Improve image resolution. What is resolution? You should also know that. So as you go through these topics, be sure that you understand the terms that I'm using in these questions, in addition to the ones on the back, because they'll show up in questions I may ask you to explain in your own words or to explain it in the context of a question.
Mm-hmm.
So refraction you should also know. But as far as theta angle and stuff like that, that's way more detail than I'm expecting. And really, as I mentioned at the top of the study guide, it's only part of chapter 3 is covered. It's really chapters 1, especially what I went over in lecture as far as the members of the microbial world. That's covered in the reading that you had for the first section of 10, 11, and 12. Those kinds of concepts will be covered in more detail than in Chapter 3. And Chapter 2, of course, will be covered in great detail as well.
Oh, he's got a couple things here.
I think
Okay.
There's
someone
the
has
ultrasound, and there's
kind
the
of a
echo.
loud
Okay. You
noise
do
in the
that, bring
background. Can you guys
those
hear
two
that?
girls up front.
Okay.
I can hear that.
You can hear it. So
All
I'm
right,
just
and
going
then you be
to,
very careful with
I
the
think,
weather, okay?
mute the person that,
Oh, got it.
let's see,
Oh, good.
try
Got Maria
muting your
there.
speakers or
Right
headphones.
here. Right.
I'm not sure if I can get rid of it. So somebody else joined us. Is Mai here with us? Who's here with us besides Gina and Mike? Or maybe who else has joined us? Can you hear? Hello? Does anyone else have questions? Am I muting myself? I'm not sure.
No, I can hear you.
Okay,
I
good.
can hear you.
Oh, good. So, sorry, did I answer that question for you, Mike, as far as the
Yes, you did.
detail?
Thank you.
Okay. Any other questions? So I'm just looking at a printout of some of the questions that came up in the feedback survey, the anonymous survey that I have you fill out each week. And I already went over in lab the questions from Chapter 1, so questions about Pasteur's experiments, and I think there were a couple questions about viruses and stuff like that. So I'm just going to, focus on this week's questions. And a few of them were about staining, and since gram stain,
I'm
even though
actually
that was one of
leaving
your
soon.
homework questions, it's not actually on
Yeah, we can
this
do that.
exam. It will be on next exam. So I'll go over the response to
I don't want to.
those questions.
No, it's okay. I don't want to bother.
Professor Oken,
Yes.
if you expand the participants window and you right-click on a person, at least when I do it, I get the option to mute and unmute. It's grayed out for me, but hopefully you as the host, you might be able to do that.
It's grayed out for me as well. I don't know why. Let's see. Is that better? Can you guys hear me?
Yeah, I think you got
Yeah.
it.
Okay. Phew. Figured it out. All right. So yeah, let me go over, so I'm not going to go over the staining questions because I want you to do the staining before I actually question you about it on an exam, so we'll go over that in the next exam. That you will be responsible for that. But I want to go over a few other questions. So someone asked about viral shedding. Viral shedding is a method of exit. So you learned about the portal of entry and the portal of exit for pathogens, and like any pathogen, whether it's bacterium or a type of fungi or parasite, viruses also have like a preferred portal of entry and a preferred and a portal of exit that they take. So shedding is just how they exit the human body. Okay, so for viruses, it's a little more confusing because there's there there are different entry and exit parts to the pathogenesis of a virus because it's dependent on a host cell to replicate. So it's always going to, if it's only in the host, if it's entered its portal of entry, but it hasn't entered the host cell, then it's not causing an infection. Okay? So it's not causing disease. It's an infection, but it's not causing disease. And then naturally after the replication, there's spread of the virus from one host cell to the next in just neighboring host cell, right? But don't confuse the entry into the body with the entry into the host cell. Those are two separate events that have to happen in the pathogenesis of a virus in order to cause disease. But then once it's released from the host cell, after it's replicated and often kills the cell or lyses the cell or releases the cell unharmed, releases a virus from the cell unharmed, it will, part of it will be shed from the body. So it will exit the body by its portal of exit. And that's shedding. Okay, so the exit from the host cell is separate from exit from our body. So that kind of has two entries and two exits for the virus.
Can I ask a clarification about that?
Sure, yeah.
Um, so it seems like it's more the method by which it exits the body, not so much like the exiting, or is it really like, let's say whatever is, used for injection, whether, depending on whatever shape and type it is, but, you could imagine like the shell of it, right? That would be what gets shed, or is it really like, because I would think what gets injected, does its thing, attacks the cell, takes over the replication, replicates itself, and then I'm thinking of the robotic looking descriptor. I forget the word. The one that looks like the moon lander or something.
Oh, the
Something like,
bacteria
is that
phage?
tends to be what gets shedded?
So in any type of pathogen, whether it's bacterial or viral, it's the entire potent form, like live bacteria or intact mature virus particle that's capable of infecting another cell is what we're concerned with as far as exiting,
Okay.
portal of exit or being shed from the body. Yep. Yeah, because all pathogens, I mean, basically they're like you've learned in biology, they're just trying to survive. So survival of the fittest. They need to find a new host, especially if they're a particularly virulent pathogen and they're killing their host. They need to get to a new host before the current host dies, or even to ensure that they can replicate again. If the host dies and they never come in contact with another susceptible host, then that pathogen is never going to be able to replicate again. So it's defeating the purpose, which is often why we see, and we'll learn, this is an idea of epidemiology that we'll talk about later. Over time, a highly virulent pathogen often kind of the symptoms and the severity of the disease dampen so that it comes into kind of a balance with where it's able to routinely and easily pass from host to host so that it can continue to replicate and exist in the environment. So hopefully that clarifies for you.
Yes, thanks.
So the next question was asking if a statement they provided was correct. The statement was the bubonic plague was a result of a zoonotic disease indirectly transmitted from an animal reservoir, in this case a rat, to human hosts via a vector, that's a flea. So that is actually correct. It's so zoonoses, as your book chapter 2 described, are these infections that humans have often become the accidental host of. These infectious diseases that result from a pathogen, whether it's a virus or bacteria, In the case of plague, it's bacteria. Um, so a type of bacteria causing disease that accidentally gets into a human host and causes disease in the human. Um, and so in the case of transmission, and this is where I asked you to pay attention to the details of the figure. So there's a figure in chapter 2 that I also have in my lecture notes, figure 210. So let me see if I can open that for us. That's chapter 3. That's chapter 1. It's covered. Let me drag one of these. 3, 1. Huh. Hold on a sec. I'm going to have to stop sharing and then open it up because it's under the...
Who's that? My goddaughter.
Okay.
I can go. I can work for you.
All right.
I told you.
Hello, did someone just join us?
You go a lot.
Suzanne?
Yes. Yes.
Hi. There's a
You're
lot of
good,
background
Joanne.
noise in your in your sound, so I'm just going to mute you for a moment, and then I'll open it up if you want a question.
That's
Okay.
fine.
Um,
I don't know.
all right, so I'm going to share the screen again now that I got the thing that I wanted to show you open. So in chapter 2, there's a figure about the transmission of disease. I'll just make this a little bit bigger. Um, okay, wait a minute. Let's see. All right. So how do I make this bigger? Oh, here it is. Okay. So this is the figure I was talking about. Just pay attention to the... What happened? There it is. Transmission. Okay. So here on the right, so there's a variety of transmissions. The most simple being direct transmission, which is human to human, and then it gets more complicated when you involve an object or a surface, which we call fomite, or contaminated food or drink or something like that, which are vehicle transmissions, so the vehicle being either the surface or the food that you consume that carries the pathogen to your body, rather than just directly through the air. And then that's an indirect method. There are a variety of indirect methods, not just vehicle transmission, but they can be zoonoses, in which an animal is carrying the pathogen and transfers it to you, or an animal to an insect to a human. And that's how the agent, whether it's a microorganism or a microbial agent, is transmitted to a susceptible human host. So there's lots of ways. The final one is directly from a parent to a offspring, and that can be from a mother to a child, for example. HIV is passed that way, for example. Okay, so so zoonoses are complex examples of transmission because they have this indirect means of getting the pathogen from somewhere to your body where it's going to cause the infectious disease. In the case of bubonic plague, it's the flea is carrying it. The flea bites the rat that for a blood meal, and during that blood meal, the bacteria is transferred from the flea to the rat, and then those that rat, then it would spread from rat to rat. But then also the flea can bite a human if the flea is carrying that pathogen, Yersinia, that causes that bacteria that causes plague, then it would make you sick. If you get a bite from a flea and it's not carrying the pathogen, then it does nothing. Let's see. Okay, so hopefully any other questions about infection cycles or disease transmission, the means, these different ways that pathogens, as far as microorganisms or microbial agents, can pass from somewhere or someone to a susceptible host. I don't know why. Let's see. I don't want to mute people and then not hear their questions, so.
I don't know. So that I know, I would love to get it. You would?
Anyone
Yeah.
anyone
So
have a
usually it's
question
all like
about
that
that?
because I'm like, you can share it. There was a
Okay. All right. So, the next question and this is also from, there are several questions from Chapter 2 this week, which makes sense. That was the challenging material. So what would happen to a person if a pathogen entered by the wrong portal? And there's no one right answer for that. And I'm not going to ask you any specific example, just understand that there is a preferred portal because the preferred portal is what brings the pathogen in contact with its attachment site. So in chapter 2 of your book, it goes over some key steps in pathogenesis, and we have a whole chapter in pathogenesis that we'll go over later, but for now, you should have in the back of your mind when we're learning about these microorganisms, and especially the examples that your book gives of pathogenic microorganisms and microbial agents, that they have certain requirements in order to cause disease. So they can infect, but they might not cause disease unless they go through these steps. And they're more specific steps that we'll learn later, but for now, you should know the basic steps. Let's see what page that's on. And that's in this section where it's talking about so section 2.1, the very first section where it's talking about fundamental host pathogen interactions. So they have to attach, whether it's a virus that has to attach to its host cell or whether it's a bacterium that has to attach to the tissue before it's able to multiply and colonize your body. Attachment is a common trait in pathogenesis that's required in order to cause disease. And then it also has to avoid your immune system at least long enough so that it can multiply and colonize. And otherwise, you won't get disease. If your immune system is able to destroy it or eliminate it from your body before it can multiply, then you have immunity. That's the goal of vaccination, is to give you immunity, that kind of boost to your immune system so that you can fight off disease before it even makes you sick. It's not that you'll never get the flu again, it's just that when the flu virus enters your body, your immune system, it can't manage to avoid your immune system. long enough to replicate and cause damage, which is seen by the symptoms that you feel when you get sick. And then the third thing is in the case of the microorganisms, they always have to be able to grow. They need nutrients, they need energy in order to grow and reproduce. So they have to obtain those nutrients. If they can't, then they're not going to be able to cause disease. So just remember preferred portal. Don't worry about if it's the wrong portal. If it's a wrong portal, in some cases it'll give you kind of a different disease. In other cases it won't give you disease. But some cases we take advantage of a different portal. So for example, we can get a flu vaccine by injecting it directly into our blood, right? And we take advantage of that portal of entry in order to create immunity and protect us from disease. But we can also use the preferred portal to get immunity, and that's by the nasal spray. That's another method of getting the flu vaccine. Let's see. Yeah. So any questions before I move on from that? Just going to. So any questions?
Not for me.
Okay. All right. Let me see. Okay, there are just a few more questions that I want to go over from the feedback. One is about members of the microbial world that we know of as archaea are often referred to as extremophiles because their habitat is either in like very hot environments or way at the bottom of the ocean where the pressure is very high, kind of environments where you wouldn't normally expect anything to be surviving. And so the question was, how do extremophiles affect humans since they're mostly non-pathogenic? And I don't have an answer for that. There's not an answer for everything. We're still just learning about these unusual microorganisms because we haven't been able to grow them in the lab, which really limits our understanding of them. But we do know that there are at least a couple that are part of our normal microbiota found in the gut. And so by their presence there in the general population, we are assuming that they have some role. We just haven't figured it out yet. And let's see. Okay, here's a question about tuberculosis. If someone tested positive for TB, had a normal chest x-ray, does it mean that the microbes remain in the incubation period of disease until the illness phase is activated and symptoms appear? And yeah, so you should know these phases of disease, and that's from, let's see, I think I have it actually in what I'm showing you in this lecture. It's probably earlier because it's from chapter 2. Yeah, here it is, stages of disease on the left of your screen. So remember that incubation phase, and there are two things that this graph shows quite nicely. One is the microbes or the pathogen, the amount of pathogen present, and the other is how our body is responding. And there's always this kind of balance we'll find in disease. It's really a fight between the pathogen's desire to reproduce and make more of itself versus the immune's response effort to limit that pathogen, limit its growth, limit its spread in the body. So the answer to the question about TB is many of us carry TB. We don't even know we have TB, but it's in our body at very low levels in this incubation phase, but we don't have any symptoms. We can't, sometimes in the incubation phase, a pathogen can be spread from person to person. In the case of TB, the bacteria that causes the infection TB is Mycobacterium tuberculosis. We wouldn't actually have the symptoms of tuberculosis in the incubation phase. In the beginnings of the prodromal, in the prodromal phase, that's when the beginnings of the symptoms would occur as a result of replication of the pathogen, and it's not until you get like what physicians will call an active phase of infection. So you're still infected even in the dormant part of the Mycobacterium TB infection. But there's an active phase and there's an inactive phase. The active phase is when you have the coughing and the very kind of phlegmy, mucusy symptoms of the infection of the disease. So you're experiencing the symptoms of the disease. And that's when you're in the illness phase. That's when there's a lot of replication of the Mycobacterium tuberculosis. It may even cause damage to your lungs that you can see on an x-ray as far as holes in your lungs or spaces of tissue death. And in the example that was given in your book in the beginning of, I think it was chapter 1, it was actually based on a real case history where a high school student in, I think it was Southern California, came down with an active case of TB, and they didn't know where it had come from, and they found that actually more than 200 students tested positive, and all of them had been exposed to it from a student that was misdiagnosed and had this persistent cough of the TB infection. So she was in that active phase, spreading the bacteria to her classmates. And any place like a school or a hospital or a dormitory is where people are going to be really close together, you know, your office at work, whatever, and they're going to spend a lot of time close together. And so that's when contagious diseases like TB are able to spread from person to person. So in the end, they had to treat about 200 or maybe more than 200 students and faculty from that school to clear their system of the mycobacterium, the bacteria that causes TB, before they got the illness or the disease. Let's see. So that touches upon these stages of disease, which you should know, as well as one other thing I wanted to point out that's on your study guide is you should understand the difference between a contagious disease and a communicable disease. So this semester we're studying microorganisms and microbial agents, and that means that we're studying infectious diseases. Okay, so we're not studying non-communicable diseases. All the diseases that we'll talk about this semester are communicable, but they're not all contagious. Contagious is person to person, so that direct transmission from one person to the next person. Okay. What else?
Professor Elkin,
Yes.
you said contagious is direct transmission only.
Well, it can also be indirect transmission,
Okay.
but really it's, yeah, it's an example. So communicable is describing indirect and direct transmission. let me see if I can find the definition for you in your chapter. Let's see. All right.
Yeah, it's not in the glossary of the
No,
book.
yeah. Yeah, I don't see it. Anyway, so all infectious diseases are communicable, but not all are contagious. It can be by indirect or direct transmission, but they're not all. So, no, I think it can be by a phone. So it has to at least have one mechanism of transmission as being direct contact. It can also have other means of transmission,
But
like,
Contagious.
the flu. So for contagious, the flu, you can get it by, shaking hands with a person, or you can get it by, uh, or you can get it by touching a doorknob that somebody just, if they had sneezed in their hand and just opened the door, so it can work both ways. But it's contagious if it's person to person is at least one of the modes of transmission. Let me see. Did I answer that question? Yes. Okay, so I just have one more question from the exit survey that I had this week that I want to go over, and it's not directly relevant to the exam, but I just want to at least answer it for the person who asked. So the question was, do you believe chimpanzees really gave humans HIV? And yeah, this is just like we were talking about before, with the bubonic plague being an example of zoonoses, or a zoonotic disease where an animal is involved in the transmission. This is another example of how the original event that led to the human to human, so for HIV, it's human to human transmission, right? But initially, there was what we call a spillover event, where from a species that was similar enough to us, that animals were involved. So that's not a zoonosis, but that's an example of how animals are relevant to infectious diseases. And zoonoses are examples of part of the transmission cycle involving animals. So something like a West Nile virus would be a zoonosis because it comes from birds, which are then, they can get sick. They're actually the in the normal host of the virus, West Nile virus, but the virus can be picked up by mosquitoes that bite the bird and then bite a human, and there's an accidental host as far as us as humans. But in the case of HIV, it's just human-to-human sexual contact by direct, so that would be direct transmission, that sexual contact that spreads the HIV. But there's a similar virus in primates called SIV, it's simian immunodeficiency virus. And it has been, so sometimes when humans are in very close contact with animals, they can get sick from a mutant form of that animal pathogen that's able to infect humans. Usually that pathogen would not be able to infect humans. So if a monkey was sick with SIV and you were like an animal care provider and you stuck yourself with a needle that had the blood from that animal, usually you wouldn't need to worry about getting sick from that animal. But in the case of things like influenza, again, there can be mutations in the microbe, in this case the influenza virus, picked up a little bit of the pig virus, the virus that, so pigs get influenza. So they picked up a little bit of the influenza virus from pigs and a little bit from birds, that also get influenza, and it combined with the right parts of the human influenza virus so that it still infected humans. Okay? So it's still human influenza, but it has little bits from other species. So there is spillover or kind of this mixing that can occur between animals that have close contact with people. These are all things that we'll talk about in more detail. So don't worry about those nitty-gritty details. Just learn the terms and the general meaning of them and be able to understand that general meaning. Okay? So now that I answered all of the questions from the feedback survey, does anyone else, I see Kiana has joined us, and some of the it just says call-in user. So please introduce yourself if you've joined us since the beginning of the review session. I'm going to go back to just my video link
Do you guys think that you'll be able to
so I can
be
see.
here
I
by
see
like
there are
8:30,
about
3 o'clock?
6 or 7 people that have joined us. Does anyone have a
I don't know if you
question?
can hear me.
Okay.
Can you
Can you
hear me?
identify yourself? Who's that?
Yeah, it's Astrid. Astrid Cruz.
Astrid. Okay, great. Welcome. Do you have a question, Astrid?
Um, if you could go over on the Pasteur's, um, no, sorry, it's not the Pasteur's. It's the postulate. How do you say that?
Oh, the Koch's postulates. Yes.
Good
Thank you for
postulate.
bringing that
Yeah.
up because I really do want to
Thank
go
you.
over that. That's
Yeah, no, I was thinking
so
this, but it can only
let
be
me
posted for
share
Tuesday at 3
again.
o'clock.
I'm not
But again, I think
sure
if we can
who
really
has
the
the
stuff
background noise. So I'm just going to take a moment to
for that box here, but you know, it's size for the food box. But the slider now. Okay.
let's see, hold on.
Okay.
I'm just trying to figure out who has the noise in the
Okay.
background so that they'll be able to
So
hear.
Rockin, from the participant menu, there's a mute all option. That might be easier than clicking individuals.
Okay. Can everyone hear me?
Yep.
Yep.
Okay, great.
Yes.
So I'm going to share my screen again because I have the study guide up somewhere here. Let's see. Here it is. So I just wanted to mention here at the top where I say open, or just below the top bullet points, open response topics, you're going to need to know that material in detail. So that's where I'm expecting you not just to kind of generally but in detail. So Koch's postulates, I don't want you to just be able to spit back the 4 steps of Koch's postulates, but also understand how they work in the context of a disease, an infectious disease, and a pathogen. So Koch's postulates are from actually, they apply to the material in Chapter 2, but I think I actually went over them in Chapter 1. Yeah, I think it's Chapter 1 that I went over them. Oh, no, here they are in Chapter 2. Okay. So on the image on the left here is similar to the one in your book. And let me just get the figure number for that so you can follow along.
I'm looking at what his cultures was. Oh, but, no, he
Okay,
doesn't.
figure 113. And figure 113, so it does actually go over Koch's postulates in chapter 1, but it goes over the 4 steps. And so the interesting thing, so these were originally developed by, forgotten his first name, but I think it's Richard Koch, or he was a physiologist, and he was interested in studying disease, and he wanted to figure out a way to figure out what was causing disease. And so he developed these 4 steps. One, you had to identify that microorganisms were involved in the diseased animal or individual, but not healthy individuals. And then you had to be able to grow up the organism, the microorganism, whether it was bacteria or fungi or some kind of parasite. It must be able to be grown, that one microorganism alone. Okay? So we call it pure culture, like no mixed. There's either a pure culture or a mixed culture. No mixed, just one organism growing, many numbers of it, but one type of microorganism. And often that meant identifying it under the microscope, and then using that pure culture to test in an experimental model, often not the host, especially in the case of human infections. We would not want to use humans as, it's just unethical to use humans as experimental hosts. So we don't do that, but we try to find an animal model for every disease that we study so that we can test to see if the same symptoms are caused. And then number 4, we need to then, once we see those symptoms, be able to isolate the same pathogen, the same microorganism causing the disease, and grow it in pure culture again. Okay, so there are several limitations, but these are the basic 4 steps. I gave you a case study to try for homework that gave an example of UTI, urinary tract infection. Urinary tract infections, the microbe in step 1, the microbe might be part of the person's anatomy. Okay, so it may be that it's a normal microbiota found on the skin in the urogenital region that gets into the urinary tract where it's not normally found, right? And causes infection. So there are many exceptions to these rules, but in general, you try to follow that, like, okay, in a healthy human, you don't have that type of microbe or that type of bacteria in the urinary tract, but in a person who's sick with a urinary tract infection, you do find that bacteria there, right? And then the organism grown in pure culture, well, that leaves out all of the non-living microbes that might cause human disease, like viruses. So viruses, sometimes you can't grow them in culture. You need to know the host cell in order to grow them in culture. So that's an added challenge with viral infections, figuring out what's the cause if it's a viral infection. And it was a challenge when in the 80s when scientists were trying to figure out what was causing AIDS, acquired immunodeficiency syndrome. But once you can get the pure culture, then you can examine it under the microscope, you can use it for testing it in an experimental model. And as I mentioned, it's not ethical to test it in a healthy human. You need to have an animal model in the case of human disease. And then recovered. So those are some limitations. There are another set of Koch's postulates for microbial agents that are modified to look for key virulent genes. So if you can't grow the microbe, you may be able to find a virulent gene that's present in that microbe that's infectious, but not in strains of the same microbe or microorganisms that aren't able to infect humans. But that's not more detail than you need to know. You need to know these 4 steps, and you need to know how they would apply to a disease, whether it's a urinary tract infection or whether it's, I think your book gives an example of West Nile virus. Those kinds of things. So are there any questions? Let's see. Astrid, did I answer your question? Or do you have other questions about those
Yeah.
Koch's postulates?
Can you hear me? Yeah,
Yes.
you did.
Okay, great.
Professor Open, I have one more, just a general one.
A general question. Go ahead.
So, in terms of how much we should rely on the study guide, if I, feel comfortable with, everything on there, or if I can get myself comfortable with everything on there, that should cover me for what? All of the exam, half of the exam. I'm not expecting you to tell me exactly how many questions, but a ballpark. Like, how much more than the study guide should we be thinking about?
So, I don't anticipate that you'll find anything that's not on the study guide on the exam.
Okay.
And I probably won't be able to ask you about everything,
Of
but
course.
at I will be asking you about these 3 topics
Right.
that are here at the top. So know those in detail. Even go ahead and, write what you know out in a paragraph so that you have some practice so that, when in the timing of the exam, when you're nervous and that's when your like mind goes blank or whatever exam nervousness you have, at least you've written something and you can start with that and just modify it to the question that I ask. Okay?
Thank you.
Any other general questions or specific questions? It's now 1:07, so I'm going to close the review session if there aren't any further questions.
That's it for me. Thanks for doing this.
Anyone else have any questions?
No, thank you.
Okay, well, thank you all for your participation. Let's see, there's one more person muted. Kiona, did you have any questions?
No, I didn't.
Okay. All right, so if there are no more questions, then I'm going to close the review session, and I'll see you guys this week in lab.
Thanks again.
Okay, bye.
Bye. Thank you.